The Hidden Faces of HIV, a scientific deep dive. Right now, over 39 million people worldwide are living with HIV. But here's what the headlines aren't telling you. While modern antiretroviral therapy has transformed HIV from a death sentence into a manageable condition, there's a silent epidemic hiding in plain sight. We're talking about brain damage, nerve destruction, and heart disease, complications that persist even when the virus is undetectable. Today, we're diving into the science that's rewriting everything we thought we knew about HIV. Welcome back to the channel. If you're new here, we break down complex medical research into content you can actually understand and use. Today's topic is critical: HIV-associated complications that affect the brain, the peripheral nervous system, and the cardiovascular system. And here's the kicker: even with successful viral suppression, these problems don't just disappear. According to research published in The Lancet HIV and multiple studies in the Journal of Neurovirology, up to 50% of people living with HIV experience some form of neurological complication. Let that sink in for a moment. So buckle up, because we're about to explore three interconnected medical mysteries and the groundbreaking research that's trying to solve them. The cardiovascular paradox. Let's start with something unexpected: your heart. The problem. Multiple studies, including pivotal research from the Journal of the American College of Cardiology, have shown that people with HIV face a 50 to 100% increased risk of cardiovascular disease compared to the general population. We're talking heart attacks, heart failure, and structural cardiac damage, even in younger patients. But why? That's the million-dollar question. The research method. Here's where animal models become invaluable. Scientists use the SIV model, simian immunodeficiency virus, which shares over 90% genetic similarity with HIV. This isn't random. It's the gold standard for studying HIV pathogenesis, as documented extensively in Nature Medicine. The experimental protocol works like this. First, researchers establish baseline cardiac function using echocardiography. Think of this as a high-definition ultrasound that measures how efficiently your heart chambers contract, how much blood they eject with each beat, and whether there's any structural abnormality. Then comes viral inoculation. Over the following weeks and months, they perform serial echocardiograms, usually every 8 to 12 weeks, tracking specific parameters like ejection fraction, left ventricular mass, and diastolic function. What they're finding? The data is revealing. Research published in AIDS Research and Human Retroviruses shows progressive cardiac remodeling, meaning the heart's structure physically changes over time. We see decreased ejection fraction, the heart pumps less efficiently; increased left ventricular mass, the heart muscle thickens abnormally; and diastolic dysfunction, the heart struggles to relax between beats. And here's the concerning part: these changes occur even when viral loads are suppressed with antiretroviral therapy. So what's actually causing this? Current hypotheses, supported by research and circulation research, point to three main culprits. 1. Chronic inflammation. HIV triggers persistent immune activation. Essentially, your immune system never fully turns off, creating a constant inflammatory state that damages blood vessels and heart tissue. 2. Direct viral protein toxicity. Even when the virus isn't actively replicating, viral proteins like TAT and GP120 can persist in tissues and directly damage cardiac cells. This has been demonstrated in cell culture studies published in Cardiovascular Research. 3. Mitochondrial dysfunction. Some antiretroviral drugs, particularly older nucleoside reverse transcriptase inhibitors, can damage the mitochondria, the powerhouse of your cells, in cardiac tissue. This is extensively documented in clinical infectious diseases. Understanding these mechanisms isn't academic, it's the roadmap to prevention. HIV-associated peripheral neuropathy. Now, let's talk about pain, chronic, debilitating, life-altering pain. The scope of the problem. HIV-associated peripheral neuropathy affects up to 35 to 50% of people living with HIV, according to comprehensive reviews in the Journal of Infectious Diseases. Imagine this: constant burning sensations in your feet and hands, shooting pains that wake you at night, numbness that makes it difficult to walk or hold objects. This isn't rare, it's one of the most common neurological complications of HIV infection. And here's what makes it particularly challenging. It can be caused both by the virus itself and by some of the medications used to treat it. We call this a double-edged sword scenario. The anatomy you need. Your peripheral nervous system is like a vast telecommunications network. It has three main types of nerve fibers. Large myelinated fibers. These are like fiber optic cables, heavily insulated, transmitting signals rapidly. They handle proprioception, knowing where your body is in space, and light touch. Small myelinated fibers, medium-speed cables, handling temperature and sharp pain. Small unmyelinated C fibers, the thinnest, slowest fibers, responsible for dull burning pain and autonomic functions. In HIV-associated peripheral neuropathy, these small unmyelinated fibers, often called C fibers, are the first casualties, and they're incredibly difficult to assess with standard nerve conduction studies. The breakthrough diagnostic technique. This is where cutting-edge research gets fascinating. Scientists pioneered a technique called intraepidermal nerve fiber density, or IENDF for short. This method, validated in multiple studies published in Neurology and Pain, works like this. A 3-millimeter punch biopsy, about the size of a pencil eraser, is taken from the lower leg, usually about 10 centimeters above the ankle. Why there? Because that's where small fiber neuropathy typically manifests first, furthest from the spinal cord. The skin sample is then processed using special immunohistochemical staining techniques. Specifically, they use antibodies against a protein marker called PGP9.5, which is found in all nerve fibers. Under a microscope, scientists can literally count the individual nerve fibers crossing the epidermal-dermal boundary. That's the boundary between your outer and inner skin. The numbers tell the story. In healthy individuals, you'd expect to see 10 to 20 nerve fibers per millimeter. Research published in Archives of Neurology established normative values adjusted for age and sex. In HIV-infected patients with neuropathy, those numbers can drop to 2 to 5 fibers per millimeter, or even lower. That's a 75 to 80% reduction. But here's what makes this so powerful. This measurement is objective. Unlike pain scales, which are subjective, IENDF gives us quantifiable data. We can measure progression. We can assess whether treatments are working. We can predict who's at highest risk.
Understanding the damage mechanisms. So how does HIV actually destroy these nerves? Research from the Journal of Neuroimmune Pharmacology and Brain has identified several pathways. Direct neurotoxicity. HIV viral proteins, particularly GP120 and TAT, can directly damage nerve cells. Cell culture studies show these proteins trigger programmed cell death in neurons. Indirect immune-mediated damage. Infected immune cells release inflammatory molecules called cytokines, TNF-alpha, IL-1, IL-6, which create a toxic environment for nerve fibers. Think of it as collateral damage from an overactive immune response. Dorsal root ganglia infection. The dorsal root ganglia are clusters of nerve cell bodies located just outside your spinal cord. Research using sophisticated imaging and tissue analysis published in Acta Neuropathologica shows HIV can establish reservoirs here, chronically damaging the nerve cells. Metabolic dysfunction. HIV infection alters cellular metabolism, impairing the energy production that nerve cells desperately need to maintain their long projections. Remember, some of these nerve fibers extend over a meter in length. They're metabolically demanding. Antiretroviral toxicity. Here's the tragic irony. Some of the drugs that save lives can damage nerves. Didonosine, DDI, stavudine, D4T, and zalcitabine, DDC, are particularly notorious. These nucleoside analogs can poison mitochondria in nerve cells, a phenomenon extensively documented in clinical infectious diseases. The good news? Modern antiretroviral regimens have largely phased out the most neurotoxic drugs. The bad news? Millions of people were exposed to them, and the damage often persists. Treatment implications. Understanding these mechanisms has led to targeted interventions. Current research, including clinical trials registered on clinicaltrials.gov, is exploring acetyl L-carnitine for mitochondrial support, nerve growth factor therapies, anti-inflammatory agents targeting specific cytokines, and topical treatments like capsaicin for symptomatic relief. The goal isn't just managing pain, it's preventing nerve damage before it becomes irreversible. HIV and the brain. Now we reach perhaps the most concerning territory, the brain. HIV-associated neurocognitive disorders. Even in the era of effective antiretroviral therapy, between 30 to 50% of people with HIV experience some degree of cognitive impairment. This is extensively documented in research from the Lancet Neurology and multiple NIH-funded studies. We categorize this into 3 levels: asymptomatic neurocognitive impairment, ANI, subtle deficits detectable only on specialized testing. The person doesn't notice in daily life, but standardized neuropsychological tests reveal problems. Mild neurocognitive disorder, MND, noticeable difficulties with memory, attention, or problem-solving that mildly interfere with daily activities. HIV-associated dementia, HAD, severe cognitive decline that significantly impairs function. This was common before antiretroviral therapy. Now it's rare, affecting less than 5% of treated patients. How HIV enters the brain. Your brain has a sophisticated defense system called the blood-brain barrier, a tight layer of cells that carefully controls what enters brain tissue from the bloodstream. But HIV is clever. Research published in Nature Neuroscience and the Journal of Clinical Investigation shows the virus uses what scientists call a Trojan horse mechanism. HIV infects immune cells, particularly macrophages and monocytes, circulating in your blood. These infected cells cross the blood-brain barrier as part of normal immune surveillance. Once inside, they release virus that can infect the brain's resident immune cells, called microglia. The virus doesn't typically infect neurons directly. Instead, infected microglia release inflammatory molecules and viral proteins that damage the connections between neurons, the synapses where all cognitive processing happens. The viral reservoir problem. Here's where it gets really complex. Even when antiretroviral therapy suppresses HIV in the blood to undetectable levels, the virus can persist in brain tissue. This is the viral reservoir problem. Research using ultrasensitive techniques published in Cell and Nature shows HIV DNA integrated into the chromosomes of long-lived brain cells. These cells aren't actively producing virus, they're latent, hiding from both the immune system and antiretroviral drugs. Why does this matter? Because these reservoirs can potentially reactivate. They may drive chronic inflammation even when viral loads are undetectable, and they're a major barrier to curing HIV. Neuroimaging insights. Advanced brain imaging has revolutionized our understanding. Studies using MRI spectroscopy reveal altered brain chemistry, decreased NAA, a marker of neuron health, and increased choline, a marker of membrane breakdown and inflammation. Diffusion tensor imaging shows damage to white matter tracts, the brain's information highways connecting different regions. PET scans using specialized tracers can detect neuroinflammation in real time, showing which brain regions are most affected. Research from Brain demonstrates that even treated patients show signs of accelerated brain aging. Their brains look structurally older than their chronological age would predict. The inflammation connection. A unifying theme is emerging from the research: persistent inflammation. Even with viral suppression, elevated levels of inflammatory markers persist in cerebrospinal fluid, the fluid surrounding your brain and spinal cord. Studies from the Journal of Infectious Diseases show increased levels of neocterin, IP10, and MCP1, all indicators of ongoing immune activation. This chronic neuroinflammation may explain why cognitive problems persist despite treatment. It's driving the search for adjunctive anti-inflammatory therapies. The interconnection. Here's what makes this truly fascinating. These three systems, heart, nerves, and brain, aren't separate problems, they're interconnected. Chronic systemic inflammation affects all three. Viral proteins circulating at low levels damage multiple organ systems simultaneously. Antiretroviral drugs, while life-saving, can have effects across different tissues. Research published in Clinical Infectious Diseases shows patients with peripheral neuropathy are more likely to have cognitive impairment. Those with cardiovascular disease show faster cognitive decline. This isn't coincidence, it's biology revealing the systemic nature of HIV's impact. Understanding these connections is pushing research toward holistic treatment approaches rather than addressing each complication in isolation. The path forward. So where does the research go from here? Biomarker development. Scientists are working to identify blood or cerebrospinal fluid markers that predict who will develop these complications before symptoms appear. Early detection means early intervention. Targeted therapeutics. Understanding mechanisms enables drug development. We're seeing clinical trials for anti-inflammatory agents, neuroprotective compounds, and drugs targeting viral reservoirs in tissue. Precision medicine. Not everyone with HIV develops these complications. Genetic studies, published in Human Molecular Genetics, are identifying susceptibility genes, paving the way for personalized risk assessment and prevention. Functional cure research. The ultimate goal: eliminating viral reservoirs, so even if antiretroviral therapy stops, the virus doesn't rebound. This would eliminate the source of persistent inflammation and tissue damage. Conclusion. Let's bring this home. HIV in 2025 is not the disease it was in 1985. Medical science has achieved the remarkable, transforming a universally fatal diagnosis into a chronic, manageable condition. But manageable doesn't mean cured, and it doesn't mean without consequences. The research we've explored today, from measuring microscopic nerve fibers to tracking subtle cardiac changes to imaging viral reservoirs in the brain, represents hope. Not empty, wishful hope, but evidence-based, mechanistically informed scientific progress. For the 39 million people living with HIV worldwide, this research isn't academic, it's personal. It's the difference between merely surviving and truly thriving. Call to action. If you found this deep dive valuable, smash that like button and drop a comment. Which complication surprised you most? Did you know HIV affected the heart and nervous system like this? Subscribe and hit the notification bell because we're continuing this series. Next time, we're exploring HIV cure research, the cutting-edge science of shock and kill strategies, and why eliminating the virus completely is so incredibly difficult. Share this video with anyone in healthcare, anyone affected by HIV,
or anyone who believes science literacy matters. Thanks for watching, and remember, stay curious, stay informed, and keep learning.